Where's Sam? There's Sam. Okay.
Talking about the good old days.
Well, those good old days. All right. Okay, it's a great pleasure to introduce, well, it's a little bit of an embarrassment too. So Srinivasa was here 8 months ago. When were you here? 6 months ago?
Last June.
What is this month? Anyway, Srinivasa years ago was a postdoc in my laboratory, having done a thesis on neural networks out of yeah, I know who you are. We know each other. Working on complex valued units. So in a network of interacting units, and you have a complex number, you get phase relationships, and you get all kinds of interesting things. So he's had an early on interest in global dynamics, and I think it's fair to say that this is an unusual introduction, by the way. I think it's fair to say that he has his own point of view on what constitutes a good brain theory. It's good to have people here like this. I think this trip, so this is by way of apology, he was sick last week and I've been in and out, and so we're hoping that he will come back again, as we're hoping Sam will do. I like this idea of having visitors come in for 2 and 3 weeks that have shared interests and want to use our toys, and then before they're eaten by the Appalachian natives, they leave. So Srini is a professor at, I'm not going to go through his scientific pedigree. You can do that. People have met him last time. But he's a professor at IIT Madras. IIT, for those of you who may not know very well, is a sure bet for getting a really good person. So in my lab, I interview anybody who applies from IIT because they have to beat so many people just to even get into the schools. And if they're in one of the premier disciplines, that's it. And he's one of those kind of people. He's interested in something that we are hoping to move into with some of the new methods that we've been developing, both ourselves and with Stephen LeConte and Brooks and Pearl in different venues, and that is recording from signals that people have not had available to them before in the functioning human brain and the epilepsy monitoring unit for the purpose of understanding sort of the other end of the problem. Neurotransmitter release, neurotransmitter uptake by glia, the production of various kinds of detectable chemicals by glia, et cetera, because we kind of believe that there's a whole missed part of the problem there that we haven't filled in for neurobiology. So he's going to share with us his ideas on the whole subject, and I think provide a good background introduction. So thanks and welcome to Professor McClure from, raise your hand, from Arizona State University, who's a friend of the family, I guess, and is here for 2 weeks. Okay, so I'm leaving today, so he replaces me. So, welcome.
Thanks a lot. Thank you. Thanks a lot. Thanks, Reid. It's always a pleasure and source of great inspiration to be around Reid and listen to him, talk to him, and all that. I was there for 1 year in his lab in Baylor, and that was like, he would say, drinking from the fire hose, right? So that in that inspiration, that impression never left me, and I thought neuroscience is the place to be, and I just stuck with it since then. So thanks to, first of all, like Virginia Tech, Carillon Center for giving me opportunity to be here. Thanks to Christine for making all the arrangements so perfectly and so on. So I want to talk about some of the modeling work we've been doing on modeling neuron-astrocyte and vessel interactions. So quickly, to give a motivation to the kind of flavor of the modeling that we were doing. So if you look at the history of neural networks and the whole connectionist evolution during the 80s and so on, so people came up with this very simple neuron model, right? All that it does is takes a bunch of inputs from other sources, applies a threshold, and it decides whether to go high or remain low. And then this is circulated, and you get, right? You have very simple learning rules, and you can explain a zillion phenomena from psychology and cognitive science, the whole PDP revolution, right? Now, so but in the 90s and especially in the 21st century, right, when people started looking at astrocytes or vessels, suddenly you never see that kind of a simplified flavor. It's always super detailed models and looking at Poisson flow in the vessels, and with astrocytes, I was shocked to find that people are counting the number of calcium atoms in an astrocytic end foot, right? I mean, where was the PDP spirit and the spirit of simplification? Where did it go? How did we miss that big, how did we deliberately choose to ignore the big picture on every occasion? So the models get so complicated, and the more complex the model, more complex the models get, the less we understand what's going on, right? And the more data we generate, the less we understand what's going on. And so I don't know, personally, I feel very uncomfortable after seeing all the beautiful, elegant theories and right of from MAR to PDP and connectionist evolution. I'm totally lost when it comes to neuroscience. I don't understand what the hell is going on. So I said, let me take a deviation, right? Search for simplified models. Right? In fact, I remember Reed saying that you need to ask question like a child, right? Why does X happen? Okay, what is the simplest explanation? I mean, these days nobody does. Nobody has time to ask questions. You're just busy collecting data, and there's mountains of data which you don't understand, but you collect even more. It's like dump yards, this right huge dump yards that you that you create, outside every big urban cities, right? I mean, you don't know what to do with all that. Data has become like that. We worship data. So I want to look for simplified models and restore that the simplicity of science, which is where the elegance of science lies. So anyway, it's a tall order, and then we're trying to do something very, very humble, very modest. So I come from this department, which is in IIT, and incidentally, IIT Madras has been ranked #1 in the country in several categories successively over the last 4 years. Okay, so if you look at neuron-astrocyte-vessel interaction, so the standard textbook picture goes something like this. So neurons are firing away and they consume energy, so they are hungry and they give out these vasoactive signals, send them to an astrocyte, and astrocyte responds with calcium elevations, and that in turn releases vasoactive substances, and which acts on the vessels. Vessels dilate and release glucose, and which is picked up by the astrocyte. Astrocyte converts it into lactate, and lactate is picked up by the neuron, which converts it to ATP, and ATP is consumed to fuel the firing activity. So this is a simple story. But although, so obviously you see that there is a feed forward and a feedback loop going on here. But if you look at functional imaging, right, this whole thing is the basis of functional imaging. People talk as if the whole thing is a unidirectional influence going from neurons to vessels. I mean, neurons are hungry and they give out this command like to the vessels, and vessels dilate and release stuff, and so there ends the matter. So because it's like a master-slave relationship. Neurons drive and vessels obey, and therefore whatever you are recording from a vessel is like basically a passive echo of neural activity, and that's how you justify, right, reading your blood volume changes in hemodynamic responses as a surrogate for neural activity. I mean, but actually, since it's a loop, we forget that very often, right? So signal can go from vessel to neuron in the reverse direction in principle. So that doesn't put vessels at an inferior position anyway. There's a mutual, it's a peer-to-peer network, right? Two networks talking to each other. So you need to have a much more complex model, a much more democratic model of neurovascular interaction. So that's what we are trying to pursue, right? Using a variety of models. So like I was saying, there are two paths, two branches to this interaction. There's one forward influence going from neuron to astrocyte, like so neuron releases glutamate, astrocyte then responds to that. There's a calcium elevation, then it releases, say, a vasoactive substance like EET. And like I want to say again, there's a zoo of molecules going on here. I'm just giving you one story, okay? So and then vessel dilates, and dilated vessel gives glucose, and glucose picked up astrocyte, converts lactate, and so on and so forth. Okay. So in fact, the reason we got into this field is earlier we did some work on skeletal muscle and perfusion in skeletal muscle. So, and that's what, so if you look at this situation, you have a muscle fiber here surrounded by a bunch of capillaries. Right now, this muscle fiber has a certain demand level. It needs so much of glucose flux per second. So, so let's assume that out of these 6 capillaries, if 3 open, right, that's enough to supply all the glucose. Now the question is, it's a hypothetical question. Which 3 will open? Which 3 will dilate? Now, so it's like, let me give a slightly feudal analogy to this. Right, I'm some rich guy and there I have 6 servants and they're willing to obey. And I say, get me 3 glasses of water. Which 3 of them will rush and go get me water? Thing is, so if everybody is too eager to please me and all 6 go, that's a wasteful kind of a service. Right? Everybody thinks other guy will go, then nobody goes, and that's bad. I'll get angry. Right? So for them to figure out that only 3 should go, they should first talk among themselves. Right? So to be able to, So to be able to dilate the exact right number of vessels, right? The vessels should talk among themselves. And not only that, vessels have spontaneous oscillation called vasomotion. So therefore, vessels should interact among themselves so that they inhibit each other. So the phases of the oscillations the vessels go out of sync, right? They show desynchronized oscillations, and that's the best condition to supply energy optimally to the muscle fibers. So these are the kinds of models that we have developed. This is published in 2007 in microvascular research. So then we ask the question, if the situation in the muscle fiber, which is generally quite passive, it's a dumb thing, just producing forces, right? It's so complicated, so interesting dynamically. What could be happening in the brain? So that's how we kind of landed into this cerebral circulation question. So one of the first things that intrigued us is, in case of a skeletal muscle, it's like the energy exchange that happens directly between the fiber and the vessels. Right fiber sends out these hunger signals, and vessels respond and release glucose and oxygen. But in case of neuron, how come there is an intermediary, this huge intermediary between neurons and blood vessels, that is astrocytic layer, or even oligodendrocyte, the whole glial layer, right? So why do we need these intermediaries between neurons and blood vessels? This was the question which we started. So to answer this question, we gave it a kind of engineering spin. Let us look at it like an engineer, and let us pose a basic requirement for delivering energy optimally to the neural tissue. So imagine that take a hypothetical situation where for every neuron there is some kind of a partner vessel. Right? Whenever this neuron needs energy, it sends a dilator, and then the vessel dilates and responds by giving energy. So there's a simple one-to-one relationship, and everybody is happy. The equation is very simple. But obviously that's not how brain works. You have lots of neurons and lots of vessels, and they're forming a network. And when a neuron sends out this hunger signals, it sprays it freely all over. It sends out through various channels, directly by reducing, for example, gases like nitric oxide, or indirectly through astrocytes and so on. So many vessels respond to that and release stuff, and that whatever is released goes through many channels to the neighborhood. Now, how do you make sure that a neuron which is working very hard, firing away at high rates, right, gets on the whole more oxygen, more glucose, where a neuron that is not working hard gets on the whole lesser glucose and oxygen? So how do you assure that? It's a very simple question. It doesn't matter what your biological substrates are, no matter what your connections are, and whether it's going freely through the ECS, diffusing through the ECS, or going through the astrocytic processes, and whatever it is. Here is a network problem which you need to answer. Because if it's not done like that efficiently, you're going to end up with very bad organisms which will be screened out by evolution. Okay, so we pose the problem like this. A neuron layer, glial layer, and vessel layer, and the input to the neurons determines their activity levels, and therefore determines their energy demand levels. The output goes to glial layer, then output of that goes to blood vessels, and feedback from vessels, glucose, and feedback from glia to here is lactate. I don't care about the actual substrate, but basically there's some kind of energy feedback coming from the glial layer to the neurons. So now for this energy delivery to be efficient, we need to achieve what I call a matching principle. That is, the total demand defined by the input into the neurons should be matched precisely, the pattern of demand should be matched precisely by the pattern of supply coming from this whole glyovascular system, which is the energy delivery system. Okay, so how do you achieve that? Now, in fact, if you think about this, this is basically an associative memory network, right? I don't need to know about the biological details. It's an associative memory network because the input given to the network and the feedback coming from all the subsequent layers are matching. That's exactly what you do in associative memory network. In fact, we call this the Boltzmann mission, right, in simple neural networks. So I saw that analogy between what is happening in neuro-astrocyte-vessel network and a simple Boltzmann mission, which we all study in your first course on neural networks, right? So Boltzmann mission works like this, or people also call it the deep belief networks. Like you have input layer and then layer 1 and layer 2, and you give a pattern to the.
Can you go back one slide and just clarify something? So why do you need is this just a cartoon?
Yeah.
Or you need some parameters that are latent, that are sort of hidden? I don't see
Yeah,
how
yeah.
to adjust them.
Yeah.
It may or may not be the glial layer.
Yeah.
Okay.
Yeah, so basically, we know from basic biology that
there's not another reason to put the glial in the middle, right? Between the vessels.
Because whatever comes from the vessel goes through glia, right? Whatever is picked up, so the other.
Yeah, I would imagine it would look more like a triangle where everybody talked to everybody.
That's true.
Okay.
That's true. Yeah, I've ignored the direct connections.
So this, as an analogy,
Yeah.
it's just so you can say, suppose there were some parameters
Yeah.
between the vessel and the neurons.
Yeah, direct ones, right? They also exist, but my understanding is the energy input from the glial layer is more than the direct inputs coming from the vessel. So I kind of ignored those.
I don't know. This is an interesting figure. I don't know what relative glycolysis
Yeah.
that goes on in the brain is in neurons versus glia.
Yeah.
Anybody know that? You know that, Stephen? You know that, John? Do the glia represent a larger fraction of the intermediary metabolism than the... Yeah, that's not known. Okay.
Okay. So even if we want to take,
I've always heard lumped together like 30% of the protein's demand, but I never heard parsed out neuron versus
Yeah, okay.
but even if you want to take that more general situation, the theory would very naturally expand to that. It's not a problem.
The theory is flexible enough to
Yeah,
completely change. If
yeah.
okay.
Yeah.
Okay, it's already looking like a bad theory, by the way.
No. No, the basic equation is still the same. Okay. Right. So this is how you define the define the problem. Right. Then so you train this. So we look at two situations. So this will give a clue as to why there should be a large hidden layer.
Wait. So what are you training it to recognize?
Yeah. So I give a pattern like on this pattern. So look at this image. This pattern is given as input to the neural layer, which is a two-dimensional layer. This board. Okay. So so suppose the whites are ones and background is 0. Right. So that means the neurons which receive ones are now firing, have higher activation levels. So they have more energy demand. Right. So the feedback from layer 1 is also a two-dimensional flux of energy. You can think of that. It's also an image. That image should match this image so that neurons, yeah. So that should happen for all the patterns. This is the requirement.
Okay, but I'm a little, okay, if I'm lost, I know somebody else is lost. So I'm a little bit lost because this looks like a Helmholtz machine.
Yeah,
Sort of
yeah, it
generative
is. Yeah,
recognition.
exactly, yeah, yeah.
Where's the energy?
Yeah, so whatever is coming back from this, I'm calling it the energy.
Why?
This is because that's what is coming back from the astrocyte layer.
But okay, so you're learning patterns of things
Yeah.
that are abstract, but have nothing per se to do with energy demand.
Right, in this version. In the next version I have.
Okay.
I'm trying to set up the big picture.
You know, I'm okay. Thank you.
Right, I'm going to build it in stages. But there are
This
problems.
is called the Helmholtz machine.
Yeah,
1996, science, hinting at all.
right. I learned it in your lab, the sleep-wake cycle and all that stuff.
Does the input image have to be binary?
No, this is just a toy problem. I mean, this is just to kind of set up. I'm just basically developing an analogy. That's all.
And okay, but these are sigmoidal units, not
Yeah.
rectified linear units.
No, yeah. In fact, no. These are actually I use oscillators because I wanted to study certain.
The Hinton et al. Paper in 2006 uses rectified linear units and sees like this vast improvement over sigmoid, right?
Okay, no, but my objective is not to look at the capacity calculations. I just want to set up the analogy between this and the neurovascular system, neuro-astrocyte-vessel. That's all. Right, to develop the analogy, give it kind of a concreteness. Right. So now the question is, why do you need this intermediate layer, the glial layer? So I look at two situations. One, there is nothing intermediate between these two, and one there's an intermediate layer. So I train them. So actually the units are not sigmoidal, not ReLU, these are oscillators, because I wanted to study the effects of synchronization of phases and all that, but I didn't have time to look at that. I really didn't explore that, develop that aspect of the model much, anyway, so I'll skip that. So these are the input images given to the network, and so the hidden layer, here there's no hidden layer at all. So feedback is pretty bad. Okay, it's not really learning the pattern much. So there's not much match between the input and the feedback. This is a feedback image when there is no hidden layer. Okay, then we added a hidden layer of size 10 by 10. So the input image is 15 by 15, the hidden layer is 10 by 10, and that's still pretty bad when we train like that. So then we keep on increasing the hidden layer, 15 by 15, it's still bad. 20 by 20, it's start looking like the input pattern, it's much better. 30 by 30, it's slightly better, but so this is the best we could get. We didn't explore, much bigger layers. So basically what is happening is, if you have a hidden layer and if you make it sufficiently big, you are able to achieve the matching condition for all the patterns. So that means as you have more and more patterns for which you want to achieve the matching condition between the demand and the supply, you need to have proportionally large glial layer. This is the conclusion, but point is it's a very super simplified model. So you can't read much biological meaning out of this, but I think it's a good start. right? So it sets up the big picture of the meaning of astrocytes in the neurovascular interaction. The whole thing rests on this idea of energy matching principle, energy demand and supply matching. So let us discuss this. What is happening here? So you have forward influence, you have neuron astrocyte. Oh, by the way, when we talk about training, we are talking about training. All these stages. So when you train, this is like simple Hebbian rule. This standard Helmholtz machine kind of a learning, right? It's a simple Hebbian rule. So during, there's a free running stage and the input, regime and so on. So when you do, what that means is all these connections are trained by Hebbian learning. No, that's slightly weird because Hebbian learning is only used to train synapses, right? The neuron to neuron connections. Whereas here I'm training neuron to glial connections and feedback connections also, and glial to vessel connections and feedback connections. They're all trained using some form of Hebbian learning, right? But it's a very simplified model. Point is, so what is the meaning that we can give to these connections? We can discuss that. Like, when a neuron is activated, what is the calcium elevation it produces in the astrocyte? That you can give it strength, like a connection strength, meaning of a connection strength to that. So on the feedback side, effect of vasodilator released by astrocyte on the vascular, on the next stage, astrocyte to vessel. When a vasodilator releases some when a astrocyte releases some vasodilator, how much increase in the vessel lumen does it lead to? You can give a strength to that, right? So that's a connection strength. Similarly, on the feedback side also you can give a notion of connection strength to these connections. Now, when we say that all these connections follow a Hebbian rule, that's a great biological hypothesis. We need to test that and see if you have LTP, LTD kind of a situation going on in all these 4 forms of connections. Each of them could be quite a momentous result if we can actually prove that experimentally. So I've been trying to convince people to take up this experiment, but so far I couldn't succeed. Nobody is interested in this. Next thing. So inputs, the connection is trainable by Hebbian learning, and the glial layer is sufficiently large. So only then we were able to get this matching condition. Now, is there any evidence for that? So first of all, is there plasticity of the NAV interaction? Now, activity-dependent plasticity of neuroglial remodeling in adult rat hypothalamus. There's a very long review paper that gives you examples of that. The notion of metabolic plasticity. Magistrati, who is a big name in neurovascular studies, he talks about metabolic plasticity, how plasticity occurs not only on the synaptic side, but in some way on the vessel side, so as to deliver energy efficiently to a given patch of tissue based on its statistics of activity in the past. But exactly how that works at cellular level, there's no recommendation for that. So here we have a theory which tells you how there can be plasticity on the metabolic side, so as to learn to supply energy efficiently to a given part of neural tissue based on its activity statistics. Then you have local density of glial cells is an indicator of metabolic demand. So that's a well-known test of metabolic demand levels of neural tissue. Then it's also known that glial-neuron ratio increases with higher environmental enrichment and stimulation. Now that's exactly what our model is predicting, right? If you want to achieve matching condition for lots of patterns, you better have a sufficiently large glial layer, and we see that in experimental. So if you grow a rat in environment and in sensory-rich environments, right, they're going to show higher astrocyte neuron ratios. So we are also saying just that. So kind of there is a lot of corroborative evidence, but you need to do some specific experiments to. So there are a lot of biological hypotheses coming out of this really toy model. There's nothing in the model. It's just an analogy. But a lot of interesting biological hypotheses are coming out of it, and we need to experiment and find out. Okay, so other areas that come out of this is if there's an interaction in both directions between neurons and vessels, how do we say that neurons are doing all the information processing and vessels are like there's a dumb pipes, right, which run fluids. And vessels also could be doing information processing. They could be having tuned responses. They could be having topographic maps. They could be having attractor dynamics. Why not? So could we think of? So in fact, we have the word like vascular computation. If you think of all that happening possibly in the vessels, right? You can think of vessels computing in the brain. Okay, so let me start with that. Yeah.
So this last question, how can you distinguish blood oxygenation level dependent responses, or people that look at birefringent responses on the cortex where they show it's a hemoglobin signal, really? Why aren't those already examples of dynamic topographic maps of the reactions of the vasculature to processing demands in the visual space and auditory space? And Why isn't that something we've already been studying for 25 years? Maybe you can help me make a distinction between the extra thing that you're claiming above and beyond the metabolic demand signals that underwrite functional imaging, for example.
Yeah, great question. So my answer to that is, so the mainstream.
What would you want? What do you want the vessels to learn? What does it make sense for them to learn?
So thing is,
Volume of tissue.
so right now whatever we read out of the vessel activity, right, we think it's just a shadow of neural activity. So you don't give any special importance to what the vessels are doing. You don't.
See, I wouldn't agree with that. I would say that it's the basis of everything everybody does here. Okay.
Which is.
Bowl imaging is a metabolic demand signal by
Yeah.
any measure.
Yeah.
Okay. Now we could quibble over whether we can identify all the contributors to it, but it is first and foremost, it's a oxygen-dependent signal, which means it's dependent on the tissue burning it down. So, like, what would you want a vessel to learn? Do you want it to learn to anticipate the next pattern that's coming through? I don't need to provide a lot of demand next, and so you see this as an anticipatory signal in the vessels?
There is data for supporting that, serotonin and DAS. All right, vessels dilate even before neurons respond to a given stimulus. So thing is, so the main.
And the evidence is that the connections onto the vessels, or some connection to the vessels, is learning that.
Yeah.
Not the glial talking to the vessels, not the neurons talking to the vessels.
So thing is, we need to tease apart.
How would you show that?
I don't know the experimental setup. I mean, I can show you the difference. The main problem is that you think of vessels as simply following neurons. So whatever they do is simply meeting the demand.
Oh, I wouldn't think that.
No, at least that's, I mean, the whole.
The reason I wouldn't think it is because metabolic efficiency has had to be hand in hand with the evolution of higher cognitive function, because if you're just a little bit more efficient than me, all things being equal,
Yeah.
In 20 generations, I'm not around.
Yeah.
So,
No.
If you were going to try to build in time dependence or time forward prediction, like that's not in your model now, right?
No, right now it's not. Yeah.
that's what I was.
What else would you have to add?
So first of all, this is only a local. It represents only local interaction. So neurons and vessels talking to each other. This whole thing is like in a small neighborhood. But the vessels also receive inputs from long-range connections from like neuromodulators, or these deep cerebral ganglia. Direct connections. Right. So that means the equation between a neuron and a proximal vessel is not very simple. So when people read out neural activity by looking at bed volume changes, that's rubbish, because what about the contribution from neurons, like, centimeters away?
Talked about this in the 90s.
Oh, really?
Saying that the vessels' responses looked anticipatory, and she wanted to credit biogenic amine synapses straight onto the vessels and all. But
so you need a more comprehensive theory. Yeah.
You take into account at all micro changes in temperature?
No.
There's been these data at hippocampus for years showing these small vascular changes coordinate plasticity and signaling pathways in the neurons as well. And I would imagine that in your model there, as you get these changes in diameter of communication, that there are micro temperature changes
Yeah.
going on. And perhaps as you think about anticipation in smooth muscle vasculature, for example, local temperature effects.
Yeah.
Having a metabolic plasticity effect, does that work its way into your model? Are there data like that plug into the model?
Yeah, in fact, there's a very old data from Casey and Milzak and Casey, if I remember right. So this is like from 60s or 70s, really old data. Like, you present as somatosensory stimulus, and you have a very slight increase in temperature, like I'm talking about like 0.01 degrees or something like that, right? So I think it's significant. It's I mean, but. And even mechanical signal, transduction plays a big role. People have this data from Filosa et al. 2016 that the vessel pressure changes of the vessel can communicate and influence astrocyte activity. Now, what do you do with that? All right. So it's really complicated. Okay. All that I'm saying is we need to look at vessels more seriously and give them kind of their own place under the sun, and not just look at them as surrogates to neural activity. And they've been given a second-rate position all along. I think we need to change that.
I don't think certainly people like me wouldn't, we're not struggling with you, we're sort of exploring how we could actually do an experiment on it, which is, I don't think we've had anything we could go measure that we thought was a strong proxy for anticipatory responses to vessels, other than
I have a couple of experiments
readouts, right?
I'll suggest at the end of the talk, 3 experiments. One of them is already proven because it's an old slide. The evidence has come last year. Okay, so, but other 2 are still open, so we can look at them.
Can I ask you a question about your previous slide, which is about the exact model? So there were, maybe it wasn't anyway. Oh yeah, this is it. So, so I'm sort of curious, like, so you say that the astrocytes are sort of this intermediate layer that's responsible for being able to reproduce any random pattern.
Yeah.
So that, which raises the question, like, what should be the relationship between the number of astrocytes and neural activity? Because if it's just, anyway, I'm sort of curious because you have sort of 2 indicators that more activity means
No, more patterns. More patterns.
that's what it should be, right? It's not purely about the amount of activity, it's sort of about sort of like the more patterns.
Yeah, so that's what the model says. If you have more patterns for which you have to achieve the matching condition.
So the first bullet point,
No, this is,
metabolic demand shouldn't necessarily matter.
no, this is from experiment.
Yeah, but this is, I'm sort of curious what you think about the relationship between glial, the number of glia, and then,
The activity levels.
yeah, but total activity level doesn't seem to matter, like the first bullet.
Yeah, but this is a very gross experiment. I mean, you just look at,
So you would predict, I think, so you would predict that if you were able to send in more patterns into tissue region 1 than you sent into tissue region 2, but you somehow normalized over the activity, tissue region 1 should have more glia in it.
sorry.
This predicts, if this, I understand your claim, filtered through Sam's question, that if you sent 5,000 patterns
With lesser
tissue region 1
average activity.
and 100 patterns into tissue region 2, but
Yeah.
somehow normed over the average activity,
Okay.
that would say it's not just some average activity level effect, but it's, I expect more glial cells.
Yeah, yeah.
More.
That's what I would expect,
If I
yes.
counted the glial cells, they'd be higher.
Yeah, that's what I would expect.
Because they're dividing.
That's what is, no, that's what is the meaning
They're
of,
migrating in, what? Where are they coming from?
they're growing, right? Yeah, they're dividing.
They're dividing.
Yeah. No,
So
that's what is meant by,
So if the glial cells are dividing, you ought to see more thymidine incorporation. I mean, you should see more DNA synthesis in this region right here. So if I send more patterns into region 1 than region 2, then I could look at DNA synthesis. That ought to go up in region 1, and I ought to be able to localize that to glia.
yeah, I mean, I don't know the biological mechanisms. I'm saying that should happen. I don't know exactly how, but
And you see the glia as accumulating evidence of these patterns. So there are
That's right.
parameters inside the glia
Yeah.
that are holding on to something about these patterns.
yeah, yeah. Yeah, so in fact,
You know Misha Aaron's paper a month ago, right?
No.
In zebrafish learning, he showed that glial cells were accumulating information about aversive events.
yeah, I won't be surprised. I'm totally with it.
No, it was like a calcium signature. Like you could show that glial cells, calcium responses were bigger than it accumulated evidence for aversive. He was shocking the zebrafish. So he's doing conditioning on zebrafish and monitoring all the glial cells.
Because at this rate, one would expect information storage to be occurring in the glial network also. And then further on in the vascular network. This is what I would bet on.
The vascular network and the glial network is there's no way to get the information really fast from one region to the other. That would be my
Fine. So they maybe they ride on the neural side. But there is stuff sitting there. You know, it's a question of
I
what.
I think these are decent ideas that have really concrete tests
Yeah, yeah,
available for it.
yeah, yeah.
Especially now when you make Tinkertoy mice.
Okay. So people said, you know, I presented this some few places, and people said, what kind of model is this? This is a toy model, and there's no biology in this, right? It's a very simple, super simplified. But because I've grown up on such models during the 90s in the connectionist ages, so I said, okay, let me do a detailed model to make some people happy. So we've done a very detailed biophysical model. So where in the neuron you have like, you know, the ion pumps and ion channels, Hodgkin-Huxley type stuff, and we wanted to have some fun. So and then it sends a glutamate signal, then you have mGlur on astrocyte, and then you have IP3 activation and calcium elevation. then EET release and acts on the vessel, and then there is glucose release and conversion to lactate, pyruvate, beta biochemistry, and the whole enchilada, right? I mean, this is the 89 equations to model like one neuron, one astrocyte, one vessel. So I hope people are happy. People are not happy. They said, no, you missed lots of other biological mechanisms. And so anyway, this is the best we could do. I mean, this is already, I think, going so far away from our basic motivation. So we studied this system, and I'll show you. So the way this whole flow pipeline works is, the first figure shows the, so the voltage level, this is, the green is spiking activity. It's spiking so rapid, so it looks like one continuous stretch. And this is the sodium nonce potential and potassium nonce potential, and then the pump currents are shown here. And this is the
This is a model from the last slide with all the
yeah. So this is just this,
all those
the results, yeah.
You have all these things in there. So you have a whole bunch of coupled differential.
89 equations.
How many?
89,
89?
just for 3 cells, 3 units.
Only 89?
People are not happy. I mean, this is what
No,
people.
that's not enough. What's wrong with you?
People consider this what people consider as serious, computational neuroscience models. I mean, I feel like puking myself, right?
I'll tell you why it's not a good model. It's just like you could get any, you could draw an elephant with that.
But this is not. People are not happy with this.
Parameters are in that model?
Huh?
How many free parameters?
I don't know. I couldn't even count, right?
You're the father of the model.
Yeah, I know. No, but I've done it because for public consumption more than for my own. So, and then this is the IP3 and calcium, and then vessel dilation. This is EET is release and vessel dilation. So you have slow oscillation of the vessel because of this.
Can I just say one thing?
Yeah.
Springboarding off of what Mike said. Mike back there, I heard his voice. Okay. Can you go back a slide? Let me just mention that. I'm sorry, they're right here. The glutamate sensing moiety, the part of the protein that senses glutamate on N-gluars,
Yeah.
is a temperature sensor.
Okay.
Did you know that?
No, I didn't know that.
Yeah. It's also a temperature, has strong homology with TRPV1 temperature sensing.
In fact, I think a good.
It might be something for you to focus on in terms of something that's kind of taking the temperature all through the tissue.
So in fact, if you want to do that seriously, like comprehensively, I think you need a real thermodynamic model of the brain.
Need a what?
A thermodynamic model, right, which handles all this systematically, right, comprehensively. But that's way beyond my, understanding of physics. But it will be nice to collaborate with somebody from thermodynamics. We already have all these ideas of applying statistical mechanics to Hopfield networks and stuff like that. That's only analogy. That's not real thermodynamics of the brain. But if you separate out the informational dynamics and thermodynamics and look at how vessels are clearing away heat from neural activity, all that stuff, and look at the informational changes, entropy changes, and it's a real hard theory, but that'll be a really grand theory of the brain.
There's a whole story that John Ullman's developed about radiating all heat and cortical size and encephalization quotients and things
Yeah.
that relate to this.
Okay. But if you want to add to that this wave dynamics, which you have begun to think about, way back, the whole idea of, right? I mean, that will be a really radical new theory of the brain. To look at, brain as a kind of electromagnetic medium, right, in which waves are propagating and so on. So, okay, so come to that. Excuse me. So yeah, the reason we took the trouble to put together such a large model is to ask this question. Is there a signal going from the vessel to the neuron? Because here you have the complete loop, right, rigged up. So now we said in this model, we just cut off the feed-forward pass, assume that there's no connection. And then we have this slow oscillations of vasomotion on the vessel side. And then you have all this feedback interactions. So what happens to the neuron? So when we have this very slow oscillation of the vessel, this is a vessel radius, right? And you see that it is modulating the neural activity. It's breaking out, it's producing some kind of a burst activity. But this is like really low, at very low energy levels. So point is, in principle, there can be a signal going from vessel to the neuron. It can modulate tamper with your neural activity. If that is the case, then, this is a simple 1 unit to 1 unit story, which is very simple. But if you can expand this over a network level, right, can you think of a situation where I inject some information into a vessel, a micro vessel, and that will have an effect on local neural activity? right? Then why do we say that neurons are the heroes and doing all the information processing, vessels are just passively obeying them? That's not this, that cannot be true, right? Anyway, so that's actually what we are gunning for in this model, and you could show that happening. So vasomotion is shown to have a reverse influence on neural firing pattern. And okay, so now, so after publishing this work, and I got a mail from Christopher Moore in Brown University. Moore has been trying to champion this idea of what he calls the hemoneural hypothesis for a couple of years, from 2008. So he said we have already been talking on these lines, but still it's a review paper. They didn't have any direct experimental evidence for that. Some interesting ideas from this review is like, so one of the problems with
What is the hemoneural hypothesis?
that there could be a signal going from vessel to neurons. Okay, it's
But to what end?
so that can be mediated by many physical mechanisms. So he gives a list of possibilities. One of them is temperature that he was talking about. So temperature change can affect neural activity through mechanisms such as latencies, right? So because the flow in the vessel removes heat and that can change temperature, so that in turn can affect neural activity. So that is one mechanism. Then there is a data from Filosa et al., which is much later in 2016, where they show that pressure exerted by a vessel can activate an astrocyte, so which in turn, in principle, can act on a neuron. So then NO release from vessels. So neurons release NO activating other synapses. You work with Gali, right, and Sajnowski. No, Edelman. So your work with Edelman and Gali, right? Yeah, okay. Yeah, I know. So then the NO hypothesis, nitric oxide hypothesis. Yeah. So just like neurons can release nitric oxide. Yeah.
I don't understand. Okay. What's the claim of the hemoneural hypothesis? You can't avoid the impact of vessels
On the neurons. And it can happen in many ways. That's all that they're saying. But it's huge controversy.
unavoidable, right?
But it's huge controversy. I mean,
Controversy.
yeah, people hate it. I mean,
They hate this idea.
yeah. And he has a lot of trouble, trying to convince people of this. And all this, for me, it looks quite reasonable. I mean, there's nothing. Okay, so I'm just citing some of the ideas from the paper, just as we're thinking on similar lines. Okay, so you saw that a very super simplified model to begin with, and then we saw a super detailed model. But then if you want to construct networks out of this super detailed model, I mean, with 3 units, 89 equations for one triplet, if you have a network of 100 units, it's going to go. So 8,000 equations, so it doesn't make any sense. So we said, okay, we asked this question. So if you have a neuron and a neurogliovascular system, what is the simplest minimal way in which I can model the influence of the whole gliovascular system on neuron? So it's like neuron is doing information processing. The gliovascular system is like a storehouse of energy and is releasing energy, right? Now, this feedback, what is the simplest way you can represent its effect on the neural dynamics? So this is a question with which we started. So, excuse me, I'm not going to go into the modeling details, but what we found is basically that energy feedback in the form of ATP, finally it all comes to ATP, right? It controls the threshold of firing of the neuron. So we did a conductance-based neuron model and the gliovascular system, and then a simplified, with the threshold, explicit threshold and all that. So we could match both behaviors by simply assuming that the energy feedback is controlling threshold of activation. So more energy, smaller threshold. It's a very simple story. So we said, okay, in future, if you want to construct large networks, certainly abstract networks of neurovascular interaction, we'll just assume that the signal coming, whatever the energy substrate coming from the vascular system, simply controls your threshold. More energy, smaller threshold. Okay, so this is what we got out of this series of models. This is one paper, whole paper where we worked it out. Okay, with these concepts in place, we propose slightly a kind of science fiction type of a model, which brings together all these different ideas into one framework in which, right, you can talk about neural vascular interaction. Now, so normally we talk about this local interaction between neurons and vessels. Neurons send out these hunger signals, vessels dilate and release energy. But that's not the whole thing that's happening. You also have these long-range connections, like there's a higher level neural regulation. So where you have this peripheral, you have this deep cerebral ganglia, like superior cervical ganglia, trigeminal nucleus, and trigeminal ganglion, and so on, which can control blood vessels in the brain. And you also have the neuromodulatory system, which can have direct terminals on microvessels. Right. So these are since these are long-range connections, if you really want to understand the equation between a neuron and a nearby blood vessel, you need to look at 3 systems. You cannot just look at 2. That is the CNS neurons, cerebral vessels, and then this what I call the vascular regulatory neural system. So there must be a high-level arbitration system which decides like which part of the brain gets on the whole more flow than other parts. If you just leave it to local regulation, no, it won't work. It's like, compare this with power utilities problem, right? If you just turn on switches, you draw power. But if everybody draws power like that, in an uncontrolled fashion, you won't be able to supply power to the entire city. You may not. I mean, if you have tight regulation on your power generation. So in India, this is what happens. So in peak summer, when there's a power shortage, there is what we call planned outages. So there's somebody at the top who decides in the utilities company, you won't get power from afternoon today from 1 to 3. This part of the city will not get power. So there must be higher level arbitration system. You cannot just leave it to local regulation. So what is that system? All these guys are candidates for being part of such a higher level vascular regulation system. So you have 3 units interacting with each other. Now this makes the picture already quite complicated, and then we need to relook at all the fMRI and functional imaging studies. If you really want to interpret it, right, you need to understand this system, and we don't really talk about them normally. Okay, so we put together this toy model. So we said, okay, let us train a simple neural network which does something very simple, like for example, an autoencoder. For somebody who hasn't heard of this, an autoencoder is a network which maps an input onto itself, right? So when you do that, and if you have a hidden layer which is somewhat narrow, right, small, it will generate the principal components of the input data set. So this is a standard textbook problem, right, where a neural network does something interesting. Okay, so now for the input and output to match, you have some notion of an output error. Okay, so that error is, we say, is computed by another box here. And this box which computes the error is like one of the neuromodulatory systems, because neuromodulatory systems are known to represent error prediction. Okay, so I'm saying this box is like represent like dopamine or something like that. Now, these neuromodulator signals go to another box which has a bunch of oscillators, and these are my vessels, blood vessels. Okay, so this signal controls the average activity levels among these oscillators, which represent blood vessels. Now, these blood vessels now talk to the hidden layer, right, sending signal in the feedback direction, right, controlling the threshold of firing of these neurons. So all the ideas we already developed them, right? So I'm just putting them all together in this network. Now I train this network. So what we found, okay, all the math, I'll skip that. What we found is when the vascular dynamics is desynchronized, right, the dynamics is rich and is desynchronized, so dynamics here. The hidden layer develops good sparse representations of the principal components of the input data. But if the dynamics is more synchronized, there's a lot of overlap between the representation. They're not really sparse like this. Okay, so the point is, here you have a network where all the components of the model are drawn from the previous discussion. And when you do that, we find that the influence from vessels to the neurons controls something about what the neural network learns. It controls your representations on the neural side. The influence on the vessel dynamics controls neural representations. That's a theoretical conclusion. Now, that's quite strange, right? So, but we need to test that. This is another thing that we can test that. But this has come about as a kind of a culmination of all the discussion that we have had so far. I think for reasons of time, I'll skip some of the other stuff. So there's a couple of studies. So we are modeling a rat whisker barrel system, right? And again, same thing. You have a neurovascular. There's no astrocyte here. It's a purely neurovascular system. And we have shown some effects on the effect of hypoxia and ischemia on plasticity. So how the barrels shrink, because of ischemia. So that can be modeled by this neurovascular model. More recently, we're still under, this is still not submitted yet. So we have the data from Prakash Kara's lab in Minnesota. So where they found that cerebral microvessels can respond to oriented bars. Okay, so that was what I was been talking about like for almost a decade now. And the last year there was a paper in Nature where they found tuned responses in blood vessels.
Can they respond to oriented bars in the absence of neural activity?
I don't know.
You could put dreads in these neurons,
Yeah.
and you could turn them
Silent
off,
them, yeah.
ask the question, and
Yeah.
turn them back
Yeah.
on.
That'll be very interesting, yeah.
He could do that experiment.
Right, so here again, we just used two networks, like a self-organizing map networks. These are what are called LISOMs. Risto Mikalainen, right, UT Austin, so he has this model called LISOM. We use just same, so it's like the same model for both neurons and astrocytes, neurons and blood vessels, vascular network. And we were able to get this result, but only thing is, one question we had, open question we had, what is the meaning of lateral interactions among blood vessels? Because for a map to work, you need to have lateral interactions. We've been talking about lateral interactions since the first study I mentioned. So here we had several options. So the late interaction, you could assume reasonably this is some kind of a center surround receptive field going on among vessels. Now the question is it on center, off surround, or off center, on surround, right? Or there's no interaction at all. So we looked at all these combinations and found that blue is off center, on surround interaction, right? And blue ones is the ones where we could get best match with the experimental data, right? If you assume that the late interactions are off center, on surround. So that's the main conclusion of the paper. So I'm still trying to catch Prakash Kara and get feedback on this model, but it's very hard to chase him and get him. Okay, the experiment I promised, right? One of the things I've been talking about for a long time is, okay, just present an oriented bar and measure response of arterioles, right, and see if they show tuned responses. That now is shown. Last year, 2016, there was a paper. Other experiment is some kind of a plasticity experiment in the astrocyte vessel connection. So just like the old Bliss and Lomo LTP experiment from the 60s, right? Something on those lines. You produce calcium activation in the astrocyte, right? And you also use microdialysis and dilate a vessel. So do it simultaneously, bang, 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 both sides. After doing it for some time, you only do calcium activation on the astrocyte side. Will there be a greater dilation on the vessel side? That's like classic LTP, if you can show that. That's a nice experiment to work on. Then another thing is to really bring home this idea of a reverse signal from vessel to neuron, right? You can repeat this well-known Newsom's experiments on motion processing, right? So you record from MT, right, of a monkey looking at a random dot pattern. And then these neurons respond for left-going dots, and these neurons respond right-going dots. Then you show left-going dots and activate the right-going neuron. It should make a mistake in judgment and should press the wrong button. And this is so now instead of activating the neurons, you activate a blood vessel, right? And can you influence its decision making? So through blood vessels, you can talk to the brain. I mean, if you can show that, so that'll be a pretty slick experiment if you can show that. I think with that I'll stop. Thank you. Yeah.
So that was an interesting talk, and thank you for being
Okay.
So here would be, I have a couple of challenges to the idea. So if I were going to guess, I would guess that you need, I like this idea that there's a specialized set of brainstem neurons that are involved in telling the vasculature, you're about to have to go do all this processing, because you don't want to ramp it up from zero. So you want it sort of, like you want a reaction, you want it at the right temperature and ready to go, and so you have a catalyst, an enzyme that does that. You think of those as separate classes of neurons?
I think there's a whole different regulatory system. If you can work it out, we can understand the stroke pathogenesis much better.
You might be able to label them by putting something in vessels that crossed over only on synapses on vessels, a label.
If you ask me, I would first do an engineering version of the model and see what are the possible scenarios. How would you even construct such a system?
Oh, I wouldn't. Those are standard, so this is a criticism. So those are standard
yeah,
models
yeah.
to kind of organize your thoughts. or
Yeah,
something.
exactly, yeah.
I think one challenge is, suppose the vessels are doing something like this and they need to learn stuff in order to match energy demand with whatever the computation is going on locally. Okay. One problem with thinking of the vessels with learning is they don't present enough surface area for all the synapses in there for the oriented bar result to be explained strictly by synapses on vessels. So Kara's results where he shows tuned vesicle responses, it's hard for me to imagine. I mean, this could just be a wrong intuition, but it's hard for me to imagine there's enough receptive synaptic surface area onto the vessels directly to account for the tuning to the bar.
Okay, but they show the tuning, right?
Well, you can have the tuning because the neurons sitting right next to it
Okay.
are tuned and have 10,000 synapses per cell. So, I mean, the vast contribution in the neuropill, the kind of synaptic neuropill there, is due to neurons, not glia or not vessels.
But like you say, if you can,
well outnumber the neurons in
Right.
terms of cells, and so they can carry their own DNA. So,
but like you say, if you can shut off neurons and then perform the experiment,
yeah,
that'll be
it's
interesting.
to try that. What kind of long distance information is not
Yeah.
available anymore?
Yes.
What kind is available?
Yes,
We want to shut them off locally
yes,
and ask what does,
Yeah.
for example, these brainstem projections.
yes,
Okay.
especially to understand stroke pathogenesis. I think if we have a good understanding of that regulatory system, we'll understand better why certain vessel is hit, when it is hit.
I think people are willing to believe this in general. I think what's been a challenge is a kind of simplifying way in.
Yeah,
This idea that you have a specialized, dedicated system to
Yeah.
anticipating
Yeah.
demand
yeah.
might be a really strong one,
And then,
especially as the cortex got bigger through evolution so quickly.
yeah. And then when the brain is compromised, parts of the brain are compromised, which parts are you willing to compromise first when you shut down brain in stages? What's the hierarchy? Like, what is the pecking order of different brain regions? If I have, like, drop my cerebral input to half, which ones am I willing to compromise first? All that,
I think there's a pecking order in the cortex.
no, in the brain on the whole, I don't know. Will you maybe preserve thalamus? Will you preserve
Well, if I
midbrain?
were, I'd keep the animal breathing. I'd keep the heart pumping.
Yeah, so that's like your midbrain centers, like your medulla and all that. Yeah. So what are the strategies? When brain is under attack, right? How would you pull back in stages? We need to understand that.
Anyway, this is a good talk. I think
Thank you.
it's a
Thank you.
challenge. I'm not,
Coming from Reed, it's right.
oh, is that right? Function of being all, just reminds me of how old I am. Anyway, thanks for
Thanks a lot.
that.
Thank you.
That was great. Anybody else got questions? No? Everybody's sitting, everybody got it? Their own ideas developed. Okay. Well, let's thank him again for coming.
Thank you very much. Thank you.